Hi, everybody. This is the last section of chapter 10. So we're going to zip through it and expect a test at some point next week, probably Friday. All right. So this is section 10.3, regulating the cell cycle. Now, the cell cycle is mitosis, which is cell division, as well as the interphase, the growth phase. So regulating cell cycles. Cells move through cell cycles at different rates. So even in human bodies, not all cells replicate or grow at the same rate. For example, muscle cells and nerve cells, or brain cells, do not divide once they're developed. Conversely, skin cells, cells in the digestive tract, and bone marrow cells do divide constantly, which is why the skin regenerates so quickly. Cells grow until they contact or bump into another cell, and then they stop growing. So think of maybe a crowded room. When there are just a few people in the room, you can move around, you can grow. But when you pack that room to the point where there are people packed in like sardines, you can't move around, or you can't quote unquote grow. So this is what happens when cells are kind of packed in tight together and they stop growing. So cell cycle regulators, regulating means kind of just putting a regulation on it, I guess. I can't really think of another word for it. So these things are called cyclins, and they regulate timing of cell cycles in eukaryotic cells to make sure that it doesn't happen too quickly, too slowly, too often, like stuff like that. So there are two different types of regulators or cyclins. So internal regulators, meaning inside, are proteins that respond to events inside of the cell. So these internal regulators are only focused on what's happening within the cell membrane or the cell wall if it's a plant. It makes sure that the cell cycle doesn't begin before chromosomes are replicated. That's a big part of mitosis. So these internal regulators make sure that all of these parts of mitosis are happening at the correct rate, happening in the correct order, and stuff like that. The opposite type of regulator is an external regulator, meaning outside of the cell. So these are proteins that respond to events that are going on outside the cell, and they can speed up or slow down the cell cycle depending on the environment or depending on what's going on outside of the cell. Uncontrolled cell growth. Now, this is what you guys are going to be doing your assignment on. Cancer. It's something that unfortunately we all know about. It has probably touched our lives in one way or another. It's a horrible disease, but it is basically just uncontrolled cell growth. It's not a disease that you catch from someone else. It's not something that you're born with. It's something that develops due to uncontrolled cell growth. These cancers form masses of cells called tumors, and they do damage to surrounding tissue. So let's say you have a bone tumor or something like that. It can not only affect your bones, but it could affect other tissues around it. So some causes are smoking tobacco. Smoking kills, so that is one huge thing. Radiation exposure and viral infections. Now, radiation is a treatment for cancer, but your whole body, when you're undergoing radiation treatments, is not being exposed to the radiation. It's super, super specifically pinpointed to just the cancer cells. So it just affects and kills the cancer cells. Most cancer cells have a defect in gene P53. And this P53 gene is what is in charge of halting the cell cycle until all the chromosomes have replicated. So without this, the cell cycle doesn't stop. If you have a defect or something wrong with the p53 gene, the cell cycle doesn't know when to stop, when to slow down. So it just replicates totally uncontrolled very quickly, and that's when cancer can grow. So here are illustrations of normal cells versus cancerous cells. So these normal cells, the blue ones on top, are nice and healthy. They're round. They have a round nucleus in the center. These are actually undergoing mitosis. You could see the cytokinesis happening. And here are the little chromosomes inside of the nucleus. And here they are, a big lump of cells. This would be called a tissue. So cancerous cells, on the other hand, they replicate very quickly. So as you could see, they're just big globs of cells. And these cells look very different from these normal cells. They aren't round. Their nuclei are not round. They're very varied in their shape and size. The nucleus is larger and darker than normal. An abnormal number of chromosomes are arranged in a disorganized fashion. So they don't have the normal amount of chromosomes that we have in all of our cells. And as you can see up here, they're nice, neat little X's. Here, they're kind of just all kind of hanging around in a haphazard fashion. These cells also don't have a boundary, as you could see. They are not these nice little neat circles with the membranes that keep their shape. They are globular and very random in their shapes. So that's how people kind of can diagnose these things as well. They can take biopsies and look at the cells to see if they look like this, if they're nice and neat, or if they look like this. That's a surefire way of diagnosing cancer. So this isn't necessarily having to do with cancer or uncontrolled growth, but it's worth discussing before you guys take your test, because some of you did have questions about chromosomes and chromatin. Are they the same thing? Are they different? So we're going to go down the line and talk about how they are different and what makes them similar. So they are both related to DNA. They are both essentially DNA. But chromosomes are tightly packaged. They're coiled like a slinky, okay? They're very coiled so they could fit in a small space. Whereas chromatin are unwound. They're kind of just like string, flying around. They are, if you took a slinky and totally unwound it and got all of the kinks out, that would be a chromatin. So chromosomes are only found in the cell during cell division. So during that 10% of time where the cell is dividing, mitosis, that is when you see these X's, these chromosomes. However, chromatin is found throughout interphase. That is 90% of the cell cycle. And as you could see in here, this is the chromatin. They are not in nice little neat X's. They are kind of just like hanging out inside the nucleus. Very stringy. And DNA is not being used for macromolecule synthesis, whereas in a chromatin, the DNA is being used for macromolecule synthesis. Big word. Okay, so that is it. That is our last slide. So you guys are going to have an Edpuzzle to do today, and that is going to be your assignment. Next week, I believe I see you guys on Tuesday. Next week, you are going to have a review, probably in the form of a Kahoot. And I will also post a crossword puzzle that you guys can do for extra credit if you want. It's not mandatory. And then Friday, you will have your test. And since I see you guys on Friday, you're very lucky, luckier than the Thursday crowd, because you have until Sunday night to complete your work for Friday. So you are going to have your test next Friday, and you will have until that Sunday to complete it. All right. So if anyone has any questions at all, please let me know. I will be holding a virtual office hour thing on Tuesday. I can't do it tomorrow because I have a meeting during the time that we're supposed to meet. But on Tuesday at 11 a.m., I will be here if you guys want a video chat with me. All right. So I hope you guys have a great weekend and get some rest, relax, stay safe.